Hello everyone. Now I am going to discuss about CNS diseases, central nervous system diseases. Okay, so in this series of videos, I would like to mainly discuss the questions which are very important for your university exams. And remember, I am not going to discuss any questions in detail. Okay, just I am going to say the hints or the salient points because CNS diseases and all you will get very less number of questions, but it doesn't mean that you can avoid it. But there is only need to remember the salient points. Okay, so in this first video, I will like to discuss a very few important questions. The number one questions, what I would like to discuss is the neuronal reactions to injury. Whenever there is an assault to the central nervous system, our body will adapt. These adaptations can be histopathologically detected. What are those reactions? The reactions of neurons to injury. Okay, so generally the reactions of central nervous system to the injury can be classified as reactions of neurons and reactions of our glial cells. We have different types of glial cells, right? Okay. Out of that, first is reactions of neurons to injury. Just remember the salient points. Okay, just understand what it is and write as per your own. Number one is actually what we call it as acute neuronal injury, also called red neuron. Red, remember, ischemia or hypoxia, hypoglycemia. What will happen? Red neurons will be produced within 6 to 12 hours after an irreversible hypoxic or ischemic insult. Red, remember, there will be indent cytoplasmic eosinophilia and there will be pycnosis and disappearance of the nucleolus. Second is called subacute and chronic neuronal degeneration. In the word itself, it is written degeneration, so which will occur chronic or subacute. That means over months to years. Fine. What are the examples which occurs in case of neurodegenerative conditions like amyotrophic lateral sclerosis, Alzheimer's disease? There will be loss of synapses. Okay, there will be loss of synapses and the neurons will undergo apoptosis. That's enough. Next is axonal reaction. The axon is mainly made up of what? We have something called as nissel substance. This nissel substance, which will undergo what degradation called as chromatolysis. Okay, this occurs, axonal reaction occurs whenever there is an assault to the spinal cord. Fine. Next is some inclusion bodies which occurs mainly following any infection, that is intracytoplasmic or intranuclear inclusion bodies, like in case of Cowdery A, Cowdery B in case of herpetic infection, Negri bodies in case of rabies infection, and owl-shaped inclusions in case of cytomegalovirus. Okay, and next is called Wallerian degeneration, which is actually a different entity, okay, which occurs following any direct assault to the nerve fiber, which we will discuss in the next chapter, another chapter. What about the reactions of astrocytes to injury? Remember, astrocytes are made up of a protein called glial fibrillary acidic protein. This glial fibrillary acidic protein is actually a type of intermediate filament. Okay, it is a type of intermediate filament. This will undergo react, that is, it will react by undergoing ramification. Okay, that is actually called reactive or gemicidocytic astrocytes. There is another type of reaction that is following hypoxia or hypoglycemia. That is nothing else but called Alzheimer type 2 astrocyte. Remember, Alzheimer type 2 astrocyte has nothing to do with the Alzheimer's disease. It is actually mainly seen in hyperammonemia, chronic liver disease, Wilson's disease, or any metabolic disorders of the urea cycle. Next is called Rosenthal fibers. This is very, very important because they are the cytoplasmic, brightly eosinophilic, irregular structures made up of two heat shock proteins, which has been asked as a previous MCQ question. What is that? Crystalline and the heat shock protein number 27, as well as ubiquitin. These are the three components of Rosenthal fibers that is mainly produced in case of long-standing gliosis. When gliosis occurs, whenever neuronal injury, in order to occupy the space, glial cells undergo proliferation in case of glial tumor and pilocytic astrocytoma. In some condition like Alexander's disease, whenever there is any leukodystrophy, Rosenthal fibers are also seen. Another important is called corpora amylase, which is nothing else but a calcific deposits. Okay, calcific deposits which are mainly made up of glycosaminoglycans, and they are mainly seen in case of pineal gland. They are mainly seen in case of pineal gland. Okay, fine. Next is reactions of microglial cells. Just remember one point, microglial cells are mesodermal in origin. They are nothing else but the phagocytes of the central nervous system. What about the other glial cells, oligodendrocytes and ependymal cells? Okay, they mainly form what we call it as ependymal granulations, and oligodendrocytes, they are mainly play a role in progressive multifocal leukoencephalopathy. There is no need that you should know everything in detail. Just remember these points. Once that is done, Now let us discuss about a very, very important topic that is called intraparenchymal hemorrhage. What do you mean by intraparenchyma? That is, the hemorrhage occurs within the brain parenchyma. So what are the causes of intraparenchymal hemorrhage? So every condition, we will discuss under the following heading. What is it actually, the definition? So intracranial hemorrhage may be like what we call as a primary hemorrhage, which occurs within the brain, or something that is called as a secondary hemorrhage. Secondary hemorrhage is what, because of some other causes, the hemorrhage occurs within the brain. For example, any bleeding disorders or any congenital disorders. Okay, that is actually called as a secondary hemorrhage. Fine. So that is actually the definition. Coming into what are the causative factors, remember the most important risk factor is actually called as the hypertension. Okay, hypertensive intraparenchymal hemorrhage. Remember, whenever you heard the word hypertension in brain, two areas where this hemorrhage occurs is nothing else but the putamen and the thalamus. Thalamus and putamen. Putamen 50 to 60%. Then comes thalamus. Okay, putamen is the area which is first to get affected in case of intensive or deep brain parenchymal hemorrhage. What are the risk factors? Hypertension, hyaline arteriosclerosis, hyperplastic atherosclerosis, and some congenital factors. What are they? Cerebral amyloid angiopathy, CAA. Okay, because of formation of amyloid peptide. Okay, for example, in case of Alzheimer's disease, we have A beta peptide. Similarly, we have some amyloid plaques, and that is responsible for some hyalinization of the blood vessels walls. That is responsible for the hemorrhage. Okay, and next another one is called the CADASIL. What is CADASIL? Cerebral autosomal dominant arteriopathy with subcortical infects and leukoencephalopathy. Remember only one thing: it is an autosomal dominant condition. What is it? A gene mutation. Notch3 gene is actually undergoing mutations. Okay, this is the important thing. Actually, coming into morphology, you can see what on gross, you can see clotted blood which will compress in the brain parenchyma. Because of this hemorrhage, there will be midline shift. That is, one half of the brain parenchyma will be shifted to the opposite side. There will be severe, cerebral edema will be there. Okay, and remember, what are they? All those, what is hypoxia, ischemic insult, what we will be seeing? Yes, we will be seeing, I have just discussed now, like acute neuronal injury. What is it? Yes, hereditary neurons will be seeing. And one more thing, what is it? Alzheimer type 2 astrocyte. These are the histopathological findings. Okay, in every condition. That is why I have already first told what are the reactions of neurons and glial cells to the injury. Just read that carefully so that you can write almost the same features, because when I will be discussing pilocytic astrocytoma, I will be again telling Rosenthal fibers. So there is no need for you to study that again. Already Rosenthal fibers, once you read from the basic, that is the first part of this video, you can remember, you can write as a histopathological finding in there. Okay, fine. That's about intracerebral hemorrhage, and in the next video I will be discussing about next of the questions.